Still getting that's good. Okay. Um, well, welcome to the annual Robert M. Doc Storm lecture. I'm Bob Mason. I'm a faculty member here in the Department of Integrative Biology and part of one of the members of the Doc Storm Committee. Um, it's a special pleasure for me to I'm not going to be introducing David Pfennig's former grad student, David Kikuchi, is going to introduce him. I'm going to tell you a little bit about Doc. But David and I were grad students together and office mates together way back at the University of Texas. But for all of you who know me, David is nothing like me. He's much more refined. He's done a lot of good stuff. So you don't have to worry about that. But I don't get to tell all my David jokes or put nasty slides in there. Let me tell you a little bit about Doc Storm. Let me put this. Oh, maybe not. Maybe I'm going backwards. Nope, I'm not doing it. Did I turn it off somehow?
Should be. Let me.
Yeah, just click it. It's only one slide. That's Doc. There we go. Or oh no, go back. I'm sorry. Now I gotta go back. There we go. All right. Sorry. All right, where we go. Okay, back on the track. Okay. All right. So known as Doc to most everyone, Professor Robert McLeod Storm, 1918 to 2017, spent over 6 decades conducting research and teaching in our department, then called the Zoology Department. Considered by many to be the preeminent scientist on Western reptiles and amphibians, Doc Storm was broadly trained and was also a respected authority on much of the Pacific Northwest vertebrate fauna. He spent a great deal of time in the field, much of that time with students. His impact is impressive and includes dozens of student co-authored publications and the mentorship of more than 70 graduate students. Doc Storm was born in Calgary, Alberta, Canada. Where's David? David, yeah, he was another one. There's secret Canadians here all the time. Before the family moved briefly to Portland and shortly thereafter to Illinois, where he was raised and went to college. He arrived in Corvallis in 1939 to pursue his master's degree in zoology and never left except for service in World War II. The title Doc is a term of endearment and respect, not for Storm's academic degree, but for his service as a medic on the beaches of Normandy. His battalion was among the first wave to land on Omaha Beach during the D-Day invasion of June 6, 1944. Surrounded by steep cliffs and heavily defended, Omaha was the bloodiest of the D-Day beaches with roughly 2,400 U.S. casualties. Troops turning up dead, wounded, or missing. Storm was awarded the Bronze Star for courage under fire while tending to the wounded. From that time on, virtually everyone called him by that name, Doc, including his wife. And so if you ever watched Saving Private Ryan, you can see that. So there's probably one of the characters is probably Doc running behind the emplacements. Okay, after the war, Storm returned to OSU in 1946 to complete his PhD with Professor Kenneth L. Gordon and was hired in 1948 as an instructor and later tenured professor in the Department of Zoology. Renowned for his pioneering research in herpetology, Doc produced nearly 50 scientific publications and several field guides on herpetology. But his most important and cherished contribution is the many students he mentored during his 36 years as an OSU professor. Doc influenced generations of biologists whose theses and dissertations deal with nearly every group of vertebrates. Famous for his open-door policy and always being available to talk with students, Doc advised 71 grad students, including 37 who completed their PhD. Those students produced at least another 50 doctorates, most of them herpetologists who made profound and important contributions to the field. Doc was also a champion for undergraduate education at OSU, winning the College of Sciences Carter Award in 1974 for outstanding and inspirational undergraduate teaching, and served as head advisor in the zoology department for many years. Doc retired in 1984, but remained active as an emeritus professor of zoology, still making many field trips to Eastern Oregon with students who loved and admired him. Doc died at age 99 in the year 2017. And so I can say anyone who's a former department head here, Virginia, could say we get emails or letters and things from former students just talking about the good old days with Doc. All right, now we're getting there. Here we go. Okay, Bill. This is Bill Lovejoy. Bill Lovejoy is the benefactor who endowed this lecture series. Bill Lovejoy was born in a small Ohio town with fewer than 1,000 people. He came from 4 generations of coal miners, so by all accounts, he should have been a coal miner, but instead he became a first-generation college graduate in 1951. After serving in the Navy, Bill earned a master's degree in geology before arriving at OSU to work on his PhD with Doc Storm. He studied mountain beavers or boomers or Aplodontia, which, so those of you, if you don't know, you better see Professor Google about that. Cool little critters, which are not even beavers at all. He earned his PhD in 1972 and then spent the rest of his academic career at Georgia Southern University, where he influenced generations of students with his teaching and research as a professor of biology. And then, quote, his quote, Doc had a rapport with students both in and out of the classroom that I tried to emulate in my teaching career, said Lovejoy. Lovejoy was one of those students who was inspired and mentored by Doc Storm, describing him as a considerate, thoughtful advisor with strong friendships and connections to many former students and colleagues. And again, quote, Doc built a strong community. He was easy to get to know, easy to work with, and well-liked, recalled Lovejoy. Inspired by his mentor, in 2016, Lovejoy established the Robert M. Storm Distinguished Lecture in Integrative Biology, an endowed series to promote excellence, advancement, and inspiration in biology, particularly vertebrate biology, to the OSU and Corvallis communities. Quote, by establishing this lecture series at Oregon State University, I hope such inspiration will continue to motivate students for years to come. We are so grateful for Bill's generous gift in honor of his beloved mentor, Doc Storm. Sadly, Bill died in 2022 at the age of 95. So you young people, like, think about the careers. You're 99, 95. I'm hoping that works for me. Okay, now I'm going to hand this off to David Kikuchi. Where's David? Oh, there he is. And he's going to introduce tonight's speaker.
All right. So it is my distinct pleasure this evening to introduce David Pfennig. So David, I know him quite well, but he comes to us from the University of North Carolina at Chapel Hill, where he has been a professor for the past 30 years. He got his start in science at the University of Texas in Austin, where he studied geology and zoology as an undergraduate and master's student before transitioning to his PhD in zoology under Jim Bull and Mark Kirkpatrick. So during his time in Texas, David and many other grad students, some of them named Bob, went on many expeditions to sample the herpetofauna of the West, a practice that resonates here with our grad students at OSU and is very much in keeping with the legacy of Doc Storm. So after his time in Texas, David did postdocs at Arizona State, the University of Arizona, and Cornell. He was an assistant professor for a little bit at the University of Illinois before moving to UNC. So this was a trip all the way from Texas over to the East Coast. But as the saying goes, you can take the biologist out of the desert, but you can't take the desert out of the biologist. So David frequently makes cross-country road trips to the playas of Arizona for his study organism, the spadefoot. And these trips often involve many members of David's lab, his wife Corinne's lab, and their children. So these outings are extraordinarily productive. The Pfennigs were featured in UNC's research stories in an article entitled The Frog Family, which chronicled their joint adventures in science. And these trips also gave their eldest daughter, Katrina, enough material to publish on a novel epigenetic inheritance mechanism while she was in high school. So beyond this, David has had a tremendous career. He has published many peer-reviewed articles, 3 books with another in prep, and was recently named a fellow of the American Association for the Advancement of Science. Among these accomplishments, David's commitment to mentorship and communicating science stands out. He has graduated 15 PhD students, but also counts as co-authors many of his undergraduates. He delivers many public lectures, such as this one, and reaches broad audiences through appearances on podcasts, BBC TV, and National Geographic. So now let's hear more from David himself. Please join me in welcoming him.
Well, thank you, David, and thank you to the committee for inviting me to be here. It's a real pleasure to have this opportunity to come tonight, and want to really thank y'all for coming. This is one of the ways I got my start in science is going to lectures like this. So hopefully there are any young kids out there, I don't turn you off. So hopefully this will in some way inspire you to go into science. So what I want to talk about tonight is I want to talk about something that I'm going to refer to as plastic life. And I want to specifically discuss how the environment can play a really profound role in influencing the development of all living things, but also their health and even their evolution. So I want to just start by describing a scenario which I'm sure we've all experienced at some point in our lives. If I can get the slides to move forward. There we are. So we've probably all done this where at some point in our lives, we stood in front of a mirror and we wondered where our features came from. So where do we get our eyes? Where do we get our well, when I had hair, where do we get our hair and our hair color? Where do we get our height and so forth? And so when we do that, we probably ascribe these features to, various ancestors. So we probably say, I got my eyes from my father, I got my hair from my mother, I got my height from my grandfather and so forth. And people do this all the time, right? And so in essence, what we're saying here is we're saying that there was something that was passed down to us. And of course, that something we always assume are genes, right? And that those genes then determine what our features are about. And so this is really the world that we live in, which is this question about do genes and the chemical that makes up genes, DNA, do they really determine our fate? And so if you go to the website of the National Institutes of Health, which is really one of the leading science organizations in the world, if you go to their National Human Genome Research Institute's website, this is what we're told on the front page. We're told that each genome contains all the information needed to build and maintain that organism throughout its lifetime. So this is what you see. And this has really led to this viewpoint in biology and even outside biology now that genes really are omniscient, right? They really do determine features. And so this was sort of encapsulated, I think, really in this book by Richard Dawkins, which he published in 1976. Which, by the way, I think is an amazing book, and I still recommend it to everybody. But in this book, Dawkins is arguing that all organisms are essentially just temporary vehicles that are built by these immortal coils, the genes, the DNA, in order to just propagate themselves, right? And so this is this perspective that sort of permeates a lot of biology. And it even sort of is going out into the sort of popular culture. You can see this little cartoon says, and you cannot change a thing as you are completely controlled by your genes. So this is the world, as I said, that we live in. Now, there are reasons for, looks like this is not working for some reason. There are reasons for believing that DNA can, in fact, control our features. And so one of the things that happened in the last century is that scientists, I'm sure many of you know, discovered what genes were made of. As I said, DNA, a nucleic acid, and they worked out the structure of that DNA and found that it consists of this double helix, sort of like a spiral staircase. And in that spiral staircase are these rungs, and those rungs are these what are called chemical bases. And there are 4 different flavors of these. They're designated by their first letter, G, C, T, and A. And another thing that scientists worked out is sort of the flow of information and how genes are read. And so they discovered that DNA is transcribed by another chemical called mRNA. It's another nucleic acid. And then that's translated into amino acids, and it's those amino acids then that make up the bodies of different kinds of living things. So for example, in all living things, if they have these 3 letters in that sequence, GAG, in nearly all living things, that will then encode for the amino acid glutamic acid. So that's true in humans, that's true in plants, that's true in bacteria and so forth. Now, one of the things that people discovered is that if you change those letters, it can have very profound influences on, in many cases, on the traits that the organisms produce. So for instance, in humans, in the gene that encodes for hemoglobin, if you change one of those letters, the A in that GAG, to a T, so from an adenine to thymine, then you change the amino acid from glutamic acid to valine, and that person is afflicted by sickle cell anemia because instead of developing normal red blood cells, they will develop sickled blood cells. So there are many stories like this, and then people discovered these things and said, therefore, genes really must be determinative. Yeah, thank you. Okay, so the problem is, though, is that if this is true, then how can individuals who have identical DNA produce very different features? So here's just one example from humans, right? So this is from a recent study in the UK where they're doing a study of identical twins, and identical twins all share the same DNA. But as you can see from just this one screenshot, they're often different in their features, right? They're different heights, different shapes of their head, and so forth. Okay, so the question is, why is that, right? Oh, thank you, Gio. Okay, so the question is why? Well, we're going to turn from humans to a small little aquatic crustacean. Oops, it's still not working. Oh, there we go. To my laptop. Okay, I'm pointing at the screen. I should point over here. So we're turning to a small little crustacean that's found worldwide in fresh water. And this is what's called a water flea. And like a lot of small things, everything tries to eat them, right? And so these guys have a little trick up their sleeve, and that is that when they are exposed to predators, they will develop this distinctive shape that you see here, where they have a spine down here, and then they'll have this sort of helmet at the top. And that's good for them because that makes them a little bit less likely to be eaten by predators because it makes them a little bigger. So what's really neat about these little water fleas is that they are clonal reproducers, right? So you can take two genetically identical individuals and you can expose some to predators and you get that distinctive form, and you don't expose other ones from that same clone not to predators. And what you see is these genetically identical individuals produce dramatically different traits, right? Different features. So what's really going on here, then, the answer to our question, of course, as many of you know, is that the environment is really critical in shaping an organism's features. So when we looked at the National Human Genome Research Institute's website, it's not true that genes contain all the information you need to build an organism. Clearly, the environment is critical. And these little organisms, as we'll see today, and others as well. So what you're seeing here then is that all traits, what biologists refer to as phenotypes, emerge during development between this interplay between genes and the environment. And this phenomenon is what we refer to as phenotypic plasticity. So broadly speaking, phenotypic plasticity is just a change in an individual's organism's features in response to changes in its environment. So we're looking at an example of that plasticity here. Now, I know many of you know about plasticity already, but for some of you, this may be a new phenomenon. And so you're probably thinking, well, maybe this just happens in these weird little things like these water fleas. But it turns out humans show a lot of plasticity. So when you work out, you develop large muscles. That's a type of plasticity. If you have light skin color, when you're out in the sun, you will tan. That's a type of plasticity. And then a type of behavioral plasticity is observed when you learn different languages. So if you're exposed to Spanish, that's a behavioral plasticity. You start speaking that language, so you differ behaviorally. If you're exposed to English, speak English and so forth. So these are all examples of plasticity. Turns out life is plastic, right? So this is a phenomenon that is really ubiquitous. It's really, I think, one of the defining features of living things. And I'm just showing you here in this slide some really spectacular examples of plasticity that I can illustrate with a slide, but a lot of forms of plasticity I wouldn't be able to show up here. So it's just things happening inside the organism, right? So the physiology might change a little bit or whatever in response to changes in the environment. But in many cases, as you can see here, they're really dramatic. So every major group of living things, from bacteria to humans, displays plasticity, at least in some of their traits. So given this ubiquity of plasticity, there are three things that I want to talk about tonight. First is, how does plasticity come about? So at the mechanistic level, what's going on? Secondly, how does plasticity impact evolution? Now, I'm specifically interested in this question because I was trained as an evolutionary biologist, and plasticity still remains very, very controversial in the field of evolutionary biology. And so we're going to talk about that a little bit tonight. And then finally, I want to just say a few words about why I think plasticity matters, why we should pay more attention to it. And so we'll just begin now with how plasticity comes about. And so for this work, I'm going to take you into the deserts of Arizona, where, as David said, I've been working now for a number of decades. And so we're going to go out there during the summers, and this doesn't happen very often these days, but we hope that the rains eventually come. And so you get these monsoonal rains, and they'll cause these temporary pools to form out there in the desert, but it'll also cause these animals to come from underground. So this is a spadefoot. They're sometimes referred to as spadefoot toads. They're actually not toads, they're actually frogs, but are really just spadefoots, right? And they're called spadefoots because they've got this little keratinized spade on the back of their foot. And they use that little spade to dig in the ground because, of course, they're living in the desert, and so they're amphibians, and so they just burrow underground and they just stay there for most of the year waiting for the rains to come. But it's the tadpoles that I've been really fascinated by, because if you go look in the ponds where these spadefoots are breeding, you find something really cool, I think, right? So a lot of the tadpoles look like the tadpoles you'd find around here. So these guys on the margins, but you also see some tadpoles that look like the one in the middle there. So they're much larger. Behaviorally, they're more active. And their shape is very distinctive as well, right? And so what we're dealing with here is we're dealing with alternative tadpole morphs, so different phenotypes, really. And so we have what's referred to as an omnivore, those smaller sort of football-shaped tadpoles. And then we've got what we refer to as a carnivore, this large-headed form. And as you might assume from their names, these two forms occupy different ecological niches. So the omnivore is really a sort of dietary generalist. They feed mostly on just the gunk that accumulates on the bottom of the pond, but also on plant material. The carnivore morph, by contrast, is a meat eater, right? So it's a predator, and so they feed mostly on these little fairy shrimp. So some of you may know these as sea monkeys that you see in the back of comic books, maybe. But they're also cannibalistic, and so they also like to eat each other. Now, one of the first things that we learned when we started studying this system is that their morphotype is strongly influenced by their diet, so what they eat. And so you can demonstrate this by just taking a young tadpole soon after it's hatched out, and you can ask, did that young tadpole eat live animal prey? So like, for instance, another tadpole or some of these fairy shrimp. If the answer to that question is no, then they will invariably develop into the omnivore morph. But if the answer to that question is yes, then many of those individuals will go on to develop into that distinctive carnivore morph. So there's, again, we're seeing the important influence of the environment, in this case, diet, and influencing what traits an organism produces in its lifetime. Now, one of the things that we also been studying, though, is, well, what's going on mechanistically? What is it about diet that can have these very profound influences on their development through their life? And one of the things that we've discovered, and other people have discovered in similar systems, is that changes in diet can really have dramatic changes in gene expression. And let me explain what I mean by this. So remember earlier we were talking about how DNA is ultimately will produce, in some cases, amino acids, right? And so that's the material that, makes up bodies of living things, right? But that's if that DNA is, transcribed and translated, as we saw earlier, into the mRNA and then into the amino acids, right? So in other words, it produces a product. Sometimes the gene is not expressed. So it means it doesn't go on to produce the RNA, and the amino acids are not produced either, right? And so what we find is that with plasticity, the environment can really determine whether or not a gene is ultimately going to produce any product. So we've been looking at this in the spadefoot toads, and one of the things we found is that of all the genes in the spadefoot genome, a whopping 35% are expressed differentially between these two different morphs. So what I mean by that is that you'll see for a particular gene, for any particular gene, it might be turned on in the omnivore and then turned off in the carnivore, or vice versa, right? So 35% of all the genes in the genome are differentially expressed between these morphs. That's really amazing, because that percentage is actually greater than the percentage of genes that are differentially expressed between different species, right? So you can make that same comparison, and it's 29% that are differentially expressed between different species. So findings like this have sort of led to an idea about how plasticity comes about at the mechanistic level. And I'm going to just step you through a little cartoon to illustrate this, right? So this suggests that many forms of plasticity might actually involve differential gene expression. So the idea is that an organism experiences some change in its environment. So think about our spadefoot tadpoles. So they either eat shrimp or they don't. Um, those can then have changes, those differences in diet can have changes at the molecular level. And so what people have discovered is that these differences in diet can cause signals to be conveyed then down inside a cell, so ultimately into the nucleus of the cell. And the nucleus is critical because that's where the chromosomes are, and the chromosomes are the material that make up the DNA, and that's where the genes are, right? So the signals get conveyed down in there, and then that can cause chemicals to bind to specific DNA sequences. And then the binding of those chemicals, those DNA sequences, can alter the expression of a nearby gene. So it's as if the gene is sort of either turned on or turned off, depending upon whether the dietary effects happen and then the changes in the gene expression happen, right? But here's the key point, is that because different environments are going to produce different signals, and because different signals affect different genes because they're going to bind to different places in the genome, what this means is that different traits can be produced from the same genome in response to different environmental signals, right? So in some cases, these chemicals, I'm just going to refer to these as chemicals now. So for those of you who know, in some cases, these might be what are referred to as transcription factors. So those are proteins that bind to the DNA to basically determine whether the gene is turned on or turned off. But one of the areas where this has been studied, and I'm going to step away for a moment from the spadefoots, another type of chemical that people have studied a lot is something known as DNA methylation, right? And so I want to just show you some of this work. And so where differential gene expression is mediated through DNA methylation. So the idea here, and this is work that's been happening over the last couple of decades, what scientists have discovered is that an environmental stimulus can cause what's called a methyl tag. And a methyl tag is simply a carbon with 3 hydrogens around it. It can determine whether or not that tag is added to the DNA strand, and then ultimately that can influence gene expression. So what happens is that when you get a lot of these methyl tags added to the DNA strand, then it causes the DNA to get coiled up really tightly. And then the genes that are in that stretch of DNA basically are essentially turned off because the other chemicals that have to come in to read those genes then it's not accessible to them. On the other hand, when there's not many of these methyl tags added, then the coils, the DNA is more relaxed, and so it's accessible to the other chemicals that have to come in to turn that gene on. So again, it's like a switch. It's like an on-off switch here. So methyl tags added, the switch is turned off, the genes are turned off. Methyl tags not added, the switch is turned on, and the genes are expressed. So let me just show you a really spectacular example of this. So you're looking at 2 mice here that are the same age, they're genetically identical to each other. So the mouse on the left, you can see is got this light color, and you can also see that it's obese, right? And you can see this mouse on the right then is sort of normal colored, and it's kind of sleek and, you know, pretty fit mouse, right? Well, the mouse on the left, its mother was not fed methyl donors when she was pregnant. So methyl donors, things like folic acid. And so what happens to that mouse then is that a particular gene known as the agouti gene wasn't methylated. And so then the product of that gene, in this case, what's called the agouti protein, was made, and that gave it this distinctive color. The mouse on the right, its mother was fed these methyl donors during pregnancy, and then that caused its agouti gene to be methylated and not to be expressed. It's basically turned off. So this agouti gene is found, by the way, in all mammals, including humans, right? So this is not just something that happens, we think, in mice. So another environmental factor, not just diet, so that can happen by it. So the mother was fed the folic acid or not. Stress can also influence methylation and thereby alter gene expression through these effects. So one of the classic studies is looking at whether or not, again, these are rodents, these are rats in this case, where researchers have found that when a pup is raised by a relaxed, high-nurturing mother, that pup then will mature as a relaxed, high-nurturing adult. By contrast, a pup that's raised by an anxious, low-nurturing mother will itself become anxious and low-nurturing. So to demonstrate how this is being influenced by methylation, what researchers can do is they'll take and this is kind of a mean experiment to do, by the way. They'll take a relaxed, high-nurtured rat, and they can inject it with a drug that adds methyl tags and convert that poor little rat into an anxious rat, right? So again, it's by adding these tags, these methyl tags to the DNA. So the bottom line here is that a lot of things can happen on top of the DNA, right, that can affect the expression of genes and thereby exert really profound influences on the traits that those organisms will produce. So we saw earlier examples of how changes in DNA sequence can have these kinds of dramatic effects on the traits organisms produced, right, through the genetic mutation, in our case of sickle cell anemia. But we also see now examples of what we refer to as epigenetic mutations, right? So you probably have all heard this term epigenetic. And what epigenetic refers to is that the epi is taken from the Greek that means on top of. So it's a change on top of the DNA sequence that is altering then the message that DNA is going to produce and ultimately the trait. And again, I want to be really clear on here. What's really critical about DNA, it's not the medium, it's the message, right? So it's the information that the DNA is conveying. It's not the sequence that's so critical, right? So if a gene is turned off, if it's masked somehow by some dominance effect, or if it's not expressed for whatever reason, it may as well not be there, right? So it's really the information with the genes that are important. That's really what we're looking at here with epigenetic mutations as well, is the information is being altered in some way. So that's a quick overview of one way that plasticity comes about. What I want to turn to next is the question that's really been fascinating me for a long time, which is really what role, if any, does plasticity play in evolution? And as I said at the outset, evolutionary biologists they've known about plasticity for centuries, right? Darwin wrote about this. Darwin's predecessors, his grandfather who wrote about evolution also basically had an idea about plasticity. So it's not a new idea to evolutionary biology, but it's something evolutionary biology sort of, sweeps under the rug, honestly. And so the question we want to ask here is how it impacts evolution. I'm going to specifically ask, how does plasticity affect the origin of new traits, right? Now, when you ask many evolutionary biologists how new traits come about, so where do they begin? Many of them will say, well, it begins with a mutation. So, whoops. They'll talk about what I'm going to just refer to as mutation-led evolution. So this is what you'll see in standard textbooks. They'll say a novel trait arises following some change in the genome. So here's a really good example. Again, we keep coming back to this example of the sickle cell anemia, right? That's a novel trait, the sickling of the red blood cells. And in this case, that novel trait was brought about by a change in the genome, by a change in the DNA sequence. But another way that that novelty can come about is through something that we refer to as plasticity-led evolution. And I want to develop this idea for you in the next few minutes, right? So the idea here is that novelty arises through plasticity. And in this case, it's when an environmental change then reveals what's referred to as cryptic genetic variation to selection. So let me explain what I mean by this. So many researchers have done the following kind of experiment, where they've taken different genotypes of some organism and they've reared them in different environments, right? And then they look at what traits those different genotypes produce, or what trait values they produce in the different environments. So we have here, say, environment 1, environment 2. This could be like hot and cold, warm and dry, whatever. And we could be looking at any, pick your favorite trait, right? And so typically what you find when you do these kinds of experiments is that you'll typically find that genotypes differ in whether and how they respond to any given change in the environment, right? So that's pretty standard, is that you'll find that those genotypes differ. But the other thing that's pretty standard is that you'll often find that in one environment, you'll find greater variation than you find expressed in the other environment. So in this case, in this hypothetical case, in this environment 2, then plasticity is uncovering what we refer to as formally cryptic genetic variation. And what I mean by that is that if you look over here in environment 1, all these genotypes are producing the same phenotype. So you wouldn't infer from that, unless you knew this from setting up the experiment, or if you sequenced them, you wouldn't even know that those are different genotypes. They would all look like the exact same genotype, right? It's only when you rear them in this environment 2, or when they experience the environment 2, when you recognize that indeed there are different genotypes here because they're responding differently to this change in the environment, right? So that's why we refer to that as cryptic genetic variation. So let me give you a real life example of this. So again, we're going to come back to the spadefoot toads. Whoops. And what we find here is that if we look at, we're taking different SIB chips here, so meaning different families, and we're splitting up in halves, and we're feeding some on detritus, And then we're feeding the other half of each SIB chip on shrimp, right? And then we're looking at the phenotypes that the offspring are producing, or the traits they're producing. And we're just using this as a continuous measure here, where more omnivore-like individuals are down here, more carnivore-like individuals are at the top, right? And so I think I've shown you something like 17 different SIB chips here. These are all from the same pond. And so one of the things that you see here, let me back up here, is that different SIB chips, different genotypes, differ in their propensity to produce carnivores. That is, they're differing in how they respond to this shrimp, right? And so what you see here is that the genotypes at the top, and I should have said each of these colored lines is a different genotype or a different SIB chip, a different family. What you see is that those ones on the top, those genotypes are exhibiting high plasticity in response to the shrimp. This one down at the bottom, though, is exhibiting much lower plasticity, right, in response to that same environmental change. So one thing this tells you that's really important is that tells us then that essentially these individuals are inheriting genes that encode different levels of plasticity. So one of the really important points I want to make here is that we don't want to think about plasticity as being a non-genetic response and non-genetic change in response to the environment, right? It's almost always got a heritable basis, and in many cases a genetic basis, right? So you often find genetic variation for whether and how individuals in the population respond to the same change in the environment. But the other important point I want to make from this, and these are real data, as I said, is that if you look at what's happening over here, the range of variation being revealed on the detritus diet versus on the shrimp diet, it's about half as much over here, right? So it's almost twice as much variation being revealed in response to shrimp than in response to detritus. Now, shrimp is actually a novel diet for most tadpoles, right? They don't normally eat shrimp. This is something these spadefoots are utilizing. But for most tadpoles, if you fed them on shrimp, it's like, what's that? You know, it's like something that's novel for them. So here we see again that novel environment revealing greater cryptogenic variation to selection. And this is really the key element of this plasticity-led evolution idea. So let me just step you through this idea with a series of diagrams, right? So we're going to begin over here in panel A, and we're going to look at a genetically variable population. So I'm showing these as tadpoles because that's what I study. And I'm showing these tadpoles as all having the same size and shape to indicate in panel A, initially they're all similar phenotypically. So they all express the same traits, right? The colors are meant to signify different genotypes, right? So you wouldn't normally see that, right? We're just setting it up that way. Now let's imagine in panel B that the environment changes. Well, typically a change in the environment is stressful for organisms, and the way they respond to that stress is by changing their phenotypes through plasticity. And so what you see here in panel B is that these different colored tadpoles now are different shapes and different sizes. So that plasticity is now being expressed. So for instance, what you see is that the maroon genotype there is becoming narrower in its body shape. The blue genotype is becoming fatter in its body shape. The green is kind of intermediate between those two. And then the orange one isn't responding at all, right? So this is that cryptogenic variation we saw earlier that's now being revealed to selection. Now, why is that so important? Why am I really highlighting this cryptogenic variation? It's critical because selection is one of the principal evolutionary forces, right? It's the force that Darwin discovered that drives evolution. Selection can only act on variation in phenotypes, right? It cannot act on variation in genotypes that are not expressed somehow in terms of changes in traits, right? So once that variation now is revealed through plasticity in panel C here, selection can start to act on it. And so let's imagine that fat-bodied form is the appropriate form to produce in this novel environment, the shaded environment. So selection can favor that fat-bodied blue tadpole, and it can act against all the other forms in there, and thereby act against the underlying genotypes, and basically, pull them out of the population. And so over time, selection might start to refine that favored form, and I'm just showing that by the tadpole becoming bigger. And eventually, you could have one of two outcomes of this process. One outcome could be you have what's referred to as a polyphenism, a novel polyphenism, where you get the fat-bodied tadpole being produced in the novel environment, and then the normal tadpole being produced in the ancestral environment, the normal environment. So that's one outcome. So that's what we refer to as a polyphenism. But the other outcome is that even if these tadpoles were to experience the ancestral environment, the original environment, they now might be fixed for producing that new form, that fat-bodied form. And that's a process that's referred to as genetic assimilation. So here we get genetic assimilation of this novel phenotype. But the point here is that regardless of whether the outcome of this process is a new polyphenism, a new form, or a new fixed form through this genetic assimilation process, regardless of which of those outcomes is realized, in both cases, you're ending up with a novel form, right? In this case, this hypothetical case, this fat body tadpole. And that whole process was set in motion, not by a change in the genome, but by a change in the environment. And that's why we refer to this as plasticity-led evolution. So we've been testing this idea with the spadefoots. And so, yeah, we've been testing it. And so we've been specifically asking, did this novel carnivore morph, did it arise through this process of plasticity-led evolution? And I'm just going to summarize the experiments. But first of all, we were set out to test two of the critical predictions of this hypothesis. And one, and this is, you do testing any theory in science, you ask, what are your predictions? One prediction is that pre-existing plasticity must have been present in some ancestral lineage. That's what got this process rolling in the first place, remember? And second prediction is that that pre-existing plasticity then underwent adaptive refinement as selection sort of honed that form in more derived lineages. So, again, regardless of whether we have a polyphenism or genetic assimilation of a novel form, we would expect some evidence of that. So just to summarize our studies here really quickly, so we've been using what's called a comparative approach. And what we're essentially doing here is comparing different lineages of spadefoots that we think might represent different stages in this process, right, of plasticity-led evolution. And so we start off over here on the left with a species, Scaphiopus holbrooki. This is the eastern spadefoot, which we have in North Carolina. And when we expose them to shrimp and we force them to eat shrimp, in this case, this one's actually eating another tadpole. You can see the little piece of a tadpole sticking out here. What we find is that there's subtle genetically variable plasticity present in that form. So we're not getting evidence of carnivore morph. We're getting a very rudimentary version of that carnivore being expressed in this lineage. And then if we look at this species, Spia multiplicata, and now we test what's happening when we feed them shrimp versus detritus, we're now getting either the omnivore morph or the carnivore morph being reduced. So we're getting evidence of a novel polyphenism present in that lineage. And this lineage moving up this family tree, this evolutionary tree, is more derived compared to that species over there. And then if we look at the most derived lineage in this three group comparison here, Spia bomifrons, what we find is that regardless of what we feed them, they're always producing the carnivore morph, right? So they're fixed, it looks like, for producing that carnivore morph. So it looks like this is, we're interpreting this as different stages in the evolutionary process, right? So we're not, we don't have a time machine here. We can't actually look at the evolution of this happening, right, in real time. We're inferring this by comparing different lineages that we suspect might be at different stages of this evolutionary process. So in other words, we think we have good support for this plasticity-led evolution hypothesis in this system. And I want to just say here, there's more and more evidence for this idea coming out now. And we've surveyed the literature and we find 40 or 50 pretty good examples where it looks like some novel trait that has arisen probably through plasticity-led evolution, right? So one of the things I want to say before I leave this topic is there's nothing here that is radical, right? This is just standard how everybody teaches how selection acts on underlying heritable variation that influences fitness, and it leads to the refinement and the potentially origin of a novel morph, right? So this is not something that's radical and that's going to overturn how we think about evolution working, right? So this is just standard evolutionary process. But let me talk about another way that plasticity might affect evolution, and this is a much more controversial way. And this is probably the reason why many evolutionary biologists just shy away from this whole phenomenon. And that's the idea that plasticity can form an alternative inheritance system which can then fuel evolution. Now, Darwin first, let me just step back for a moment. Darwin first argued that for evolution to occur through natural selection, you simply have to have three conditions satisfied in a population. First of all, he said you had to have variation, meaning that individuals have to differ somehow in their characteristics. And we talked about this earlier. Remember, that's why we said the cryptic gyration is so important, because the plasticity was revealing phenotypic variation. So in this hypothetical example, we've got these insects that are different colors, right? Secondly, he said you had to have differential survival or reproduction of individuals in that population because of their distinctive characteristics. So let's say in this hypothetical example, these birds are preferentially eating the green insects and leaving these orange ones alone. And then thirdly, he said you had to have some way of passing those favored characteristics on to the next generation. In other words, you had to have some mechanism of inheritance. Now, when Darwin was developing this theory, he didn't know about genes, even though he and a guy named Gregor Mendel, who was kind of the father of genetics, were actually contemporary. They were living in different parts of Europe. Darwin didn't know about Mendel's work. Mendel knew about Darwin's work, but Darwin didn't know about Mendel, right? So Darwin said, if you have those three conditions satisfied, then you're guaranteed to get evolution by natural selection. In this case, the frequency of these different colored bugs changing over time, right? Now, we saw earlier how inheritance can be critical, right? So we talked about inheritance and plasticity. But what I'm going to be talking about in this next section is I'm not talking about the inheritance of genes that encode for plasticity, as we saw earlier in the spadefoot example. Rather, we're talking about the inheritance of the specific plasticity response itself, right? Now, again, this is a really controversial idea. Many of you probably are familiar somewhat with it, and it's often attributed to a 19th century French scholar named Jean-Baptiste Lamarck, right? Now, Lamarck was a big fan of the idea that plasticity can really exert influence on populations. He really talked a lot about use and disuse. And this is a quote, this is translated from the French, where he says, all that has been acquired or changed in individuals during their life is transmitted to new individuals who are related. So in other words, he was really big on this idea that by using a trait or by not using a trait, you could cause it to become better developed, or you could cause it to become more poorly developed and to ultimately atrophy. And so he said, this is why, you know, moles that live underground, you know, lose their eyes and so forth, right? Because they're not using their eyes. And this is why he said some shorebirds develop really long legs because they're staying in water all the time, and so their legs get longer, right? But then he added this second point about once those features are acquired in a lifetime, they're passed on to the next generation. And people ascribe that to Lamarck so much that it's sometimes referred to as Lamarckian inheritance. But I want to be really clear here. That's an idea that goes back to the ancient Greeks, right? So Lamarck was just taking this idea that features acquired in an individual's lifetime are passed on to the next generation. He was just taking it as a given. So it's unfortunate this is referred to as, I say, Lamarckian inheritance. But in any event, most modern biologists will argue that the so-called inheritance of acquired characteristics does not work, right? And so you still hear this all the time, and you read this in textbooks, right? Where they say, stay away from Lamarck, right? This does not work. But there are very clear-cut examples where features acquired in an individual's lifetime can in fact be passed on to their offspring. So in other words, features that are acquired via plasticity can affect traits in the organism's offspring. And the reason for this is that living things have evolved several ways to pass material and information to their offspring. And that's really what inheritance is about. Inheritance is about passing material or information to the next generation. We've gotten to the point where we define inheritance as the transmission of DNA or genes, but there are other ways that organisms can pass material or information on to their offspring. So clearly they can do so through chromosomal genes. I mean, there are many, I'm not going to deny that. There are many clear-cut examples of that. But parents can also pass to their offspring characteristics that shape their lives. Sorry about this. They can also pass their offspring epigenetic factors. They can pass symbionts like microbes. They can pass body traits like condition or size. They can pass ecological traits like burrows and nests. They can also pass on behavior and culture to their offspring, right? Now, why am I emphasizing this? Well, all of these different characteristics or all these different factors here, these non-genetic factors can all be triggered by a change in the environment. So they essentially constitute what we refer to as transgenerational plasticity. So I want to just give you an example of this, and I'm going to focus first on the transmission of epigenetic factors and how this can affect offspring and how it could potentially mediate evolution. And then I'm going to end by talking about transmission of behavior and culture and how that can also be passed to offspring. And I'm going to give you an example where that is clearly shaping the evolution of the organisms that we'll be talking about here. So let's begin with what we refer to as transgenerational epigenetic inheritance. So one of the early examples of this really focused a lot on these methyl tags that we talked about earlier. So recall from earlier that the environmental stimulus can determine whether or not a methyl tag is added to the DNA. And remember, that can influence gene expression and thereby influence the traits that the organism is producing. Well, it turns out that there are enzymes that can recognize these methylated sites on the parent strand of the DNA during DNA replication, and then it can basically add them to the daughter strand. And so what that means then is that methylated the status of the DNA then is inherited by the offspring, right? So again, this is a controversial idea. It doesn't happen all the time, but there are clear examples where this is happening, right? So what this means then is that in this way, the altered state of a gene and any features that are affected by that gene then are passed down to the offspring. And so this is happening even in the absence of a change in the DNA sequence. So here's one of the early examples of this. So again, involving rodents, these are rats where researchers exposed a pregnant female to pesticides and found that her sons, her grandsons, and her great-grandsons suffered poor health and reduced fertility. And they also found there were altered DNA methylation in the sperm, right? And so there's just every day there are new examples of these where changes in the environment are causing changes in some epigenetic factor like DNA methylation, and then those are being passed down through at least a number of generations, right? Let me show you now an example of cultural inheritance where a change in behavior was acquired in an individual's lifetime, and then that was passed on to the offspring, and that indeed is even shaping the evolution of that group of organisms. And here we're talking about white-throated sparrows, and there's a really cool example in white-throated sparrows where they recently underwent a dramatic change in their call. And so what we see here is a new song arose recently in the white-throated sparrows, and it's basically swept across the entire continent. So let me explain what it is. So here we are before 2000, and these little red dots are showing you what's called this triplet call. I'm going to play what this call sounds like. So many of you are probably familiar with that call, right? But then around 2000, a new call appeared in Western Canada. And that new call is what's referred to as the doublet call. And that new call now is swept all the way across the continent. So I hear this new call now in North Carolina. It's like, what's that? You know, I had to use my Merlin to figure out what the bird is that's causing that, right? It is this new call that is just, you know, in just less than 20 years, moved all the way across the continent. Now, what does this have to do with plasticity? Well, in songbirds, like these white-throated sparrows, they learn their songs, and they learn them early by listening to and imitating adult tutors, typically their father, although not always. So what this means then is this is an example of transgenerational plasticity. So here we see an example where a plasticity that can be passed on to the offspring is mediating the evolution of an important trait, in this case, the call these birds make. You know, you wouldn't see such a really rapid change in the features of a population like this normally through a change in the DNA sequence. So here is something that's happening potentially within the lifetimes of individual birds, where it's just sweeping across the population so dramatically. And as I said, there are other examples of this as well that we could talk about. So let me just end by just talking briefly about why I think plasticity matters. Well, you know, plasticity, we think, is really going to probably be important in helping organisms cope with rapidly changing environments. So new traits, as we've seen today, produced through plasticity, they are frequently beneficial. They are not always, but frequently they are. They also tend to occur in many individuals simultaneously. That's in contrast to a genetic mutation, which is going to be random with respect to whether it's helpful for the organism in that environment, but it's also only going to occur in a single organism and then potentially its descendants, right? But here, if a change in the environment's happening, it's going to affect many individuals simultaneously in a population. But also, as we just saw with the case of the birds, they can spread through a population within a generation. So, you know, evolution is by definition a change in the characteristics of a population, inherited change in the characteristics of population across generations. So by definition, evolution can only happen from one generation to the next. So it's always going to be happening across generations. Plasticity can happen within generations. So what this means is that it potentially can happen in real time, much, much more rapidly than an evolutionary change. Now, why is this important? Well, it may be important because it may be decisive in reducing the risk of a population going extinct when they're experiencing really dramatic and rapid changes in their environment. So let me give you a couple examples of this. So one example comes from bird species, and this is a recent study that showed that if you look at the innovation rate across different types of birds, so some birds are really smart and they really innovate, and I think Marslove talked about this at his Storm talk last year, right? Some are kind of dumb and they're not innovating as well. And if you relate that to what their risk of extinction is, so we got least concern over here and we've got critically endangered over here, you see an association between the innovation rate, the behavioral plasticity, and the extinction risk. So those that are more innovative, that display higher levels of plasticity, are less likely to go extinct than those that are less innovative, that express lower levels of plasticity. But let me show you another example, and this comes from the work of my new colleague, Eric Rydell at UNC, and he's been studying salamanders in the Southern Appalachians in Western North Carolina. So one of the really cool things about North Carolina, I'll brag here a little bit about my home state, is that it is the salamander capital of the world. I know you got some amazing salamanders here in Oregon too, but it's kind of not, you guys are kind of like second place, right? So Western North Carolina, in some places, the state of itself, which is a much smaller state than Oregon, it's got like 70 species of salamanders. And most of those are in the western part of the state. Essentially, every mountain range has its own species of salamander. So Eric has been focusing on this red-cheeked salamander. It's in the genus Plethodon, the lungless salamanders. And what he's been asking is, how important is plasticity to allowing these organisms to survive in what we anticipate is going to happen to the climate in the Southern Appalachians for the next 50 years? So he's basically developing what's called a biophysical model. So he's asking how good are they at being able to, because they're lungless, so they basically breathe through their skin. But the problem with breathing through your skin is then you lose all your water, right? So the question is, what's the energy cost to basically living in an environment that's going to become hotter and drier, right? And so he's modeling this when there's no plasticity, and this is their current range. And the sort of cooler colors mean that their energy balance is more favorable. The hotter colors means it's less favorable. But then he models it with the physiological and behavioral plasticity. So behavioral plasticity is things like knowing to get underground when it's really dry. Physiological plasticity is basically shutting off the skin so that they don't lose as much moisture. And so what he finds is that with plasticity, the net energy balance is much more favorable over a wider geographical range. So we feel pretty good about these guys, that as long as they keep their current levels of plasticity, they might be able to deal with what's about to come down the pike, for into the next few decades, right? And so what are the implications of this? This means that maybe as we focus our efforts on what species we should worry about, maybe we should be worrying about those like those dumb birds, right, that don't have much plasticity, that are going to be really at risk, right? So organisms like these salamanders that have really, well adapted plasticity might be less risk. Okay, so the other thing I just want to conclude with, though, is that plasticity also has implications for health. And again, the health scientists, we have a really good medical school at UNC, and I go to talks over there. They understand this stuff. They get it. I mean, they're talking a lot about plasticity. And so recall earlier that we saw how diet and stress can have really important influences that can last the individual's lifetime, right? So we saw examples from the spadefoot toads. We also saw examples from the mice. Well, it turns out that these effects can also sometimes be passed to descendants through these sort of non-genetic means that we talked about earlier. So let me just end by kind of a creepy story of involving in humans, right? And so this is a recent study that analyzed harvest records from a town in northern Sweden. You can see there from 1874 to 1910. And what they found is there was a strong association between food availability in one generation and disease in later generations. And specifically, what they found is the amount of food a grandfather had between the ages of 9 and 12 was especially important. Now, in humans, that's the age when boys form the cells that are going to produce the sperm, between that age of 9 and 12. So this is what they found. So if the grandfather had low nutrition when he was a boy, his sons and daughters, his grandsons and granddaughters had normal health. So nothing to see here. But if that grandfather had high nutrition when he was a boy, again, his sons, his daughters were fine, his granddaughters were fine, but his grandsons have a much higher risk of cancer than the boys that had low nutrition, right? So we don't know what the exact effect is here, right? So it's probably not DNA methylation like we talked about earlier. It's probably another not epigenetic mechanism. It's probably something called non-coding RNAs that are being passed down. But the point here that I'm making, it could be something else even. The point here is that what you do in your lifetime matters, right? It matters not just for your health, but it potentially matters for the health of your descendants, right? So you're going to pass more than just, if you have kids, you're going to pass more than just your genes to future generations. You're potentially going to pass these epigenetic factors and other kinds of non-genetic factors like what we talked about earlier to your descendants. And that's important to keep in mind. So just to sort of wrap up here, the bottom line is plasticity is a normal part of development. It's not some freak show. It's something that all organisms show. It can impact evolution. So it can have these really important consequences over evolutionary timescales. But it also has really, I think, important practical applications. So it matters for the preservation of biodiversity. It also really matters for health as well. So I want to just close also by just thanking you all for the invitation. But I also want to, since this is a public talk, I also want to thank you for your support of science funding. So all the work that I talked about today that was conducted in my lab was all funded by the National Science Foundation. And a lot of the work, the rodent work that I talked about, was also funded by the National Science Foundation or the National Institutes of Health. And so as many of you know, these two funding agencies are under a lot of threats these days. And I think it's going to be really unfortunate, right, if that threat is sort of realized, right? So a lot of the important discoveries, I mean, some level, who cares about tadpoles or whatever, right? So, I used to go talk to ranchers out there and they'd all go, why does this matter, right? And so it matters because we don't know where this is going to lead, right? It might lead us to new discoveries about how the environment shapes the traits that organisms produce and how those traits might be passed on to future generations. So again, thank you all for the invitation, and I'll be glad to take any questions.
We just have, before we do questions, people often run out at the end of questions. So we would like to take a moment to present David with a token of gratitude in the form of a highly plastic spadefoot. And we would also like to invite the audience to examine the bottom of your seats to see whether you may have won a miniaturized version of that plastic spadefoot. Also, check vacant seats nearby. Okay. So save those until later. If you found them, save those until later. You can pick them up outside after we finish. All right. So yeah, we're ready to field questions now.
Well, thank you very much. Great talk. Surfing the web the other day, I found out that there are people that sell themselves as epigenetic coaches nowadays, and they teach you how to activate and deactivate your genes. And I found that frustrating, to be honest. Maybe you've shared the feeling. I was wondering, you mentioned a little bit about the health implications at the end of your talk, and I was thinking when you talk about the rats that were anxious versus relaxed, I was thinking, oh my God, I should be I have to be a relaxed father, otherwise my daughter will be anxious. I was wondering, what for the future, what do you feel like the future next steps will be of epigenetics, both in terms of applying to science in the sense of experimenting with organisms, but also to health?
Yeah. Thanks. I guess I'm in the wrong business. Epigenetic coach, that sounds great, right? Sounds like a really good gig. I mean, look, I think and I want to be really clear, I am not some kind of nut who says DNA and genes aren't important, right? Clearly, it is the oldest inherited system that life has evolved, and it's a very effective one. I mean, a couple of years ago, the Nobel Prize was won by the person who sequenced the Neanderthal genome. That was using DNA that was over 30,000 years old, right? So DNA is really, really efficient at transmitting information, and it lasts a long time, right? So these epigenetic factors aren't going to potentially last for 30,000 years. I think the really important point is to say that, we need a more comprehensive perspective on how, how traits come about and how inheritance works, basically. And so again, I don't think I'm saying anything here that people wouldn't recognize, from a long time ago, so we need to get rid of silly quotes like that thing I showed you from the National Human Genome Research Institute website, where they're saying, the genome it conveys all the information needed to build that organism and maintain it throughout its life, because we clearly know that's not important. And so that sort of lets people off the hook in terms of, well, people might think I've just got good genes, so I don't need to worry about what I'm going to do in my lifetime. And maybe I'm going to mess myself up, but my kids inherited my good genes and all my good money or whatever, and so they're okay, right? And we know clearly that's not the case. And so I think that's really where we need to get. As I said, my sense is the biomedical field is getting there. There's still some people who, geneticists, I think, are slow. It's really evolutionary biologists that I think are having a lot of the really serious challenges, with that idea. So yeah, good question. Yes.
Yeah, I can take the mic. Thank you. So I'm really interested in polymorphisms, and so I was curious about the tadpole system that you're working with, where you have this omnivorous morph, and this carnivorous morph that is, they're very structurally different. Are there intermediates in that range between them? And if so, do you know the factors that are resulting in, increased carnivory and
So there are intermediates in this system. So we do get intermediates in the wild. Those intermediates get weeded out though really quickly in their lifetime. So they get weeded out by a process known as disruptive selection that's acting. So where selection favors individuals that are in most ponds, not every pond, in most ponds, selection favors really extreme carnivores or really extreme omnivores. And the reason for that, we think, is because, carnivores have characteristics that allow them to basically be really good predators on things like fairy shrimp and other tadpoles. Omnivores can't do that because they don't have the big jaw muscles and the other traits. And then conversely, omnivores have features that enable them to be really good at just eating gunk off the bottom of the pond or plants. Those are the two main niches in these ponds. There's no sort of intermediate niche. So if you're kind of like in between those two, you're kind of like a jack of all trades, but master of none, right? And selection tends to not favor those. So yes, you can produce them in the lab, and if you just raise these in the lab, you'll get lots of intermediates. You put them in a natural pond in the wild, and you'll find those intermediates just start disappearing in within about a week, right? So you'll just not see them in the population. And we've gone out in many natural populations, documented that. And most natural populations, at least in Arizona, are experiencing very strong, again, disruptive selection. And many people for a long time thought disruptive selection was rare, but our study and others like it have shown that disruptive selection is probably more common than we previously thought. Yeah, so that's a good question. Oh, I'll let you. Okay. I'm just gonna get my exercise.
Thank you. So you show those two different phenotypes or morphs for the juveniles. Is there any persistence into adulthood in that? Do we see any differences with the adults at all?
So we don't know. So I suspect we've not found any obvious differences, right? So we found some differences in the adults. But part of the problem is that in order to do those studies, what we do is we take known carnivores and omnivores, and then we take them in the lab and we rear them under common conditions. And when they reach sexual maturity, which in this system, males get there in about a year, females at about a year and a half or 2 years. You got to wait a while. We're not seeing any differences, right? But the problem is we're rearing them under common conditions in the lab. In the wild, the carnivores are coming out faster than the omnivores. So the environment's gonna be different for them, right? And we don't know fully where they go in the desert. For all we know, they hang out over here and the omnivores are all hanging out over there, right? So there may very well be differences, but there are theoretical reasons for not expecting there to be differences carrying over in the adult phase. And that is that there's theory that suggests that, selection can sort of favor these kinds of polymorphisms when it can do it at one life stage and not affect the other life stage. Because you could think about this as carnivores and omnivores are just different strategies to become a frog, to get out of a pond before it dries up and to grow really fast and get out of that pond and to get big. And then you're all the same as adults, right? And so that may be what's going on here. We just don't know. But yeah, that's a great question. You had a question here.
So speaking of exercise, would you consider that to be environmental factor during aging where it has such profound effect on people's health?
I'm sorry, could you develop that a little bit?
Well, the exercise, we're constantly told that during aging, if you exercise, then one is more healthy.
Yeah.
Then, so say two siblings, one even twins, one would exercise and the other not. Would you consider that this is part of plasticity?
I would. I mean, and I'm not saying anything very profound. We all know that, right? And so that, I mean, exercise changes your physiology. It also changes your morphology. I mean, make an example, exercise in early life, we're not born with a kneecap, right? So the kneecap is something that develops through exercise, basically, when you're really tiny, right? And so similarly, as you get older, if you're exercising more, you're just maintaining that muscle mass, for example, which can be critical for, keeping you upright. You're also maintaining your cardiovascular system. And again, if you don't exercise, those change, right? That's plasticity, but they can change for the worse. So yeah. 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 And we do accumulate, just as we age, we accumulate genetic mutations in our cells. We also accumulate epigenetic mutations. There have been many studies that have looked at that. So going back to those identical twins, people have looked at the genome, and they're I said they're identical, but everybody, there's what are called somatic mutations do occur, but they've also looked at the epigenome of identical twins, and by looking at gene expression, and as they age, they start becoming more and more different, right? Because their environments are different. So yeah.
Thank you very much for your talk. I loved it. So I'm applying things that you have just told us to the topic of biodiversity conservation. And the conundrum is we have limited resources for conservation, but sometimes a unique population is identified, salamanders with big heads, dark color. And we know that those are phenotypes that can be plastic in different species. Do we afford those unique populations conservation resources to maintain a single site? Or do we take resources and, look at the rarity of species overall? And I think I have learned something from you, and the answer is yes.
Mm.
What do you think?
Well, when E.O. Wilson, who's the person who coined the word biodiversity, when he first coined that term in his book, 1992, he defined biodiversity as the total inherited variation of all living things, right? He didn't classify biodiversity measuring it by number of species, right? So getting at your point, biodiversity is just that variation which you see in different populations. It could be carnivore, omnivore. It could be like you said, the big-headed salamander, or the dark, or whatever. And so, I mean, this is a little bit beyond me, right? But I think it's important to preserve that biodiversity at that level, because that could be really critical to ecosystem functioning and stuff like that. I think that going to something that I was trying to say at the end, I think that one of the things where plasticity becomes important for conservation practitioners is understanding that maybe we should be focusing our efforts on organisms that don't display a lot of so-called adaptive plasticity, because they're probably the ones that are going to be in a lot of trouble. The other thing, though, I think we can say is that we need to start understanding better what the cues are, which I didn't really talk about tonight, but what the cues are that induce plasticity. And we need to make sure we're not screwing up the organisms by messing with those cues. So for instance, many of y'all know the story of sea turtles, where they will mistakenly eat, plastic bags because they mistake them for their jellyfish, right? So that's a type of plasticity gone bad there, right? And so when we start messing with those cues, we're gonna take what was an adaptive response to say, hey, something that looks like that, something to eat, and I'm gonna do well by eating that. And knowing that when we mess with those cues, we're gonna really mess things up, right? And so that's, I think, another application of that. And this is where we need to get out of sort of academia and get out there and start talking to the people on the ground, and giving them some advice on, don't ignore plasticity. Let's make it work for us. Let's make it work for the organisms that are at the greatest risk. So I think that's a really important frontier of plasticity research, is applications of biodiversity preservation. Yeah, thank you for asking that.
Let's give David another massive thank you for that fantastic talk. Thanks, Joe. Thank you. All right, so yeah, as you're filtering out, we have some display tables out. You're welcome to interact with them. And David may field a few questions, yeah, as we're on our way out the door.
Gio, thank you.